Hello everyone, welcome to chapter 5, part 1. In this chapter we are going to be talking about histology, which is the study of different tissues. Now, just to have like a brief little intro, in our previous chapter, chapter 3, we talked about cells, how they are the fundamental unit of life, and how all organisms are composed of at least one or more cells, and of course that all cells come from pre-existing cells. Now, what we're going to do in chapter 5 is we're going to see what happens when you take cells and you bundle them together, and what we see happening is that collectively these cells will then start forming tissues. Now, our body is made out of trillions of cells, and there are all types of different cells. You have cells that make up your skin, you have cells that make up your stomach, your small intestines, you have your specialty cells in your nervous system. So there's a wide variety of cells that we see happening. And these cells collectively can be bundled together into 4 groups when we start looking at tissues. And those are going to be your epithelium tissue, your connective tissue, which is the most numerous, the nervous tissue, which you exclusively found in the nervous system, so we're thinking brain, spinal cord, and then last but not least, our muscular tissue, which is going to be making up our muscle category, such as our skeletal muscle, our smooth muscle, and our cardiac muscle. Now, part of what we have to do in chapter 5 is we are going to have to discuss what makes each category different from the other, and how will you be able to identify them, and more importantly, can you list some examples of where you can find these tissues within your different organs or organ systems. Now, you want to keep in mind that oftentimes when you discuss an organ, you will see that it's composed of multiple types of tissues. This is important to point out, especially for those of us who are also taking the lab. When you grab a slide, for instance, of the kidney, you're going to see that there's going to be a wide variety of different tissue types. You will have a little bit of epithelium, you'll have a little bit of connective tissue that's mixed together, and part of what we have to figure out in A&P is how can we distinguish one tissue type from the other, and that will take a little bit of practice and a lot of patience. So my goal in chapter 5 is to introduce you to these 4 categories of tissue, and also show you how you can figure out which one you're looking at and what some of the specific characteristics are for the groups. So without further ado, let's go ahead and let's dive into histology, which is the study of tissues. Now, first things first, depending on how the tissue is prepared will determine the best views that you're looking at. So part of what you'll see, especially in those of us who are in lab, is that when you start studying your different slides, the slides will have indications that will tell you if they were produced due to a cross-section, an oblique section, or a longitudinal section. These sections indicate how the tissue was cut, and depending on that, it will overall give you a different view of the item. So let me give you an example of that. Let's take a look at the illustration that's on our particular slide right here. And as you can see on the top, on the left-hand side, you have a long bone, and then you have a little cylinder that's on the right-hand side. And the first option is if we take our samples and we make a longitudinal section. And that basically means that we're going to cut it in half from top to bottom. So you can see the little glass plane that they've inserted, so they can show you exactly where the cut is. And then interesting enough, if you follow the black arrow, you're going to be seeing what part of the specimen you're able to view. Now, how about we take the same exact bone and cylinder, and now we do a cross-section. A cross-section is when you cut from left to right, across the abdominal section, and as you can see, now you have more of a circular view of the specimen. Completely different than the one that the longitudinal section provided us, which was more concentrated in a length style from top to bottom. Now, the third option is if we make an oblique section. This usually involves that you make about a 90-degree angular cut as you're going through it, so more of a diagonal cut. And when you do that, notice once again how now you're getting oblique sections, hence the name, of the specimen. Now, the reason I like to point out this figure is to kind of indicate to you that as we start tackling histology, you're going to see that not all slides are created equal. Not all of them will show you the same view because it all depends on how it was prepared. And also, it's up to you to kind of explore these slides from multiple angles, just so you can get more practice and a better understanding of how these cells interact together to form the particular set of tissues. Now, without further ado, let's go ahead and start taking a look at our first category of tissue, which will be our epithelium tissue. All right, I do want to mention something real quick. In our exam, when we take our exams for our course, there will be no illustrations or pictures on the exam. Instead, you're going to have pictures of wanting to identify the different tissue types. Those pictures or illustrations will be found in your homework assignments. In your exam, what I'm going to be asking you is what is the location of these particular tissues, and I'm also going to be asking you some of the specific characteristics that set one tissue apart from the other. So speaking of some characteristics, let's go ahead and let's start off with epithelium tissue. Epithelium tissue, most of us will think along of skin because epithelium tissue is excellent for covering surfaces. It usually involves multiple cells that are closely interacting with each other. Usually you'll have an upper surface that is exposed to either the environment or to an internal space, and then you'll have a bottom surface that will interact with another tissue type below it, most likely connective tissue. Epithelium tissue is also known for the fact that it is avascular. Avascular means, A means without, vascular is for the blood vessels, so it means it does not have any blood vessels. And this is going to be really important because in our next chapter, we're going to take a look at our skin and we're going to discuss why our skin keeps shedding and there's a constant need to produce new cells. Part of that will have to do with the avascular ability of the epithelium tissue. Now, obviously, tissue cells are a fundamental unit of life, as discussed previously, so their nutritional demand will be based on having access to blood vessels in the connective tissue that lies underneath them. Another item about epithelium tissue, as I was mentioning to you before, is that it will often entail the cells closely intermingling and connecting with each other. And by doing so, they're going to be creating sheets of cells, which are one of the unique characteristics of epithelium tissue. These cells tend to have a high rate of mitosis, and also what we see happening is that when we take a look at their position, there will often be what we like to call a basement membrane, a basal surface, and an apical surface. Now, since the epithelium tissue tends to be the covering, what we see happening is that below it, it will interact with connective tissue. The layer between the epithelium and the connective tissue is called the basement membrane. Just like you would have a basement in a house, it's the bottom layer. That bottom layer of epithelium cells that comes into contact with the connective tissue underneath it is going to be called the basement membrane. And this is then where you have your basal surface, which is the surface of the epithelium cells facing the basement membrane. The other end of the epithelium tissue will often be exposed to the environment or to an open surface. That end is going to be called the apical surface. Now, don't worry, in a little bit I'll show you some illustrations and I'll clearly highlight for you the basement membrane, the basal surface, and the apical surface. Now, how exactly do we go about naming epithelium tissue? Well, you're going to see that when you look at the names, it's going to tell you two things. It's going to tell you the individual shape of the cells, and it's going to tell you how they are organized. Now, let's start at the bottom right here under B. When it comes to the cells, we're going to have three options. The cells will either be considered squamous, cuboidal, or columnar. Squamous cells, as you can see from the illustration here, they tend to have more of an abstract shape. Most books will describe them as fish scale-like. They are not very consistent in their overall structure. Whereas if you compare the next cell, which is a cuboidal cell, it will look more like a cube, but more likely under the microscope when you're looking at an actual slide, it tends to look more like a circle. Either way, it will have a very consistent shape, whether it looks like a box or a ball. Whereas the squamous cell type is very abstract in shape. Each of them can have a slightly different arrangement than the other. Our third option is probably the easiest cell to identify, and that is called the columnar cell. And as the name indicates, these are going to be cells that have a lot of cytoplasm, and it's going to look like an elongated cell, the shape of a little rectangle or a column in the 3D version. So you have either a squamous cell, which means it's abstract, fish-like and fish scale-like. You have a cuboidal cell, which means that it looks like a little cube, a little circle, very consistent in its shape. And then you have your elongated cell, which is the columnar cell. Once you figured out what shape the individual cells are, remember epithelium tissue is all about cells interacting with each other. The next thing you want to do is how exactly are they arranged? So what is the class of epithelium? Your first option, and this is going to be under A, is that you have what we call a simple arrangement. A simple arrangement means that you just have one layer of cell. So here on that illustration, you can see that the cells are nicely lined up together, all in one layer. Now, notice how the cells are columnar in shape. Notice how one part of the cell is interacting with the bottom part. This is your basement membrane. And notice how one part of the cell is exposed to an external surface, the environment. That's the apical surface. Your second option is if you now have cells that are layered together. So you have two or more layers stacked on top of each other. That's going to be called stratified arrangement. So stratified arrangement, I'm going to take my cells, I'm going to line them up in multiple layers. So you can see I have one layer here, there's my second, my third, my fourth. They're stacked on top of each other. Once again, I can see I have a basement membrane and I have an apical surface where it's interacting with an open environment or open space. And then my third option, this is one of my favorite ones, it's called pseudostratified. Pseudo means appearing as if. So it's going to appear as if it's stratified, but it's just one layer of cells. So take a close look at the illustration and find your basement membrane. So here's your basement membrane. And then what you're going to notice is that every single one of the cells that has been drawn connects to the basement membrane. And because every single one connects individually to the basement membrane, that means that it's really one layer of cells. However, it looks like it's multiple layers because the cells all have different shapes, different sizes, and oftentimes it looks as if they are just condensed together. They're just packed in too heavily, so they don't always reach the apical surface. See how the small ones right here don't reach the apical surface, the top surface? So that's why it's going to appear as if it's stratified, but it's really just one layer. So we call that arrangement pseudo-stratified. So now that you have become, hopefully, somewhat familiar with the individual shapes of the cells as well as their arrangement, we're going to go ahead and combine those two features so that we can name our different epitheliums, because the name of the epithelium tissue will tell you its shape as well as its arrangement. So for instance, if I come back here, I'm trying to erase some of this so we can have a clear picture again. If I come back here to my first class, my simple class, I can see it's simple because it's one layer, and I can see that my individual cell is a column. So what I'm going to do is I'm going to combine those, and the official name of this tissue would be simple columnar epithelium because it's one layer and all of them are columnar. If I come over here to my right-hand side, these are my stratified layers because it's multiple. If I look at the individual cell shape, they look like little cubes to me. So this one is going to be called stratified cuboidal epithelium. So notice how when you're naming your epithelium cells, you're going to tell me their name of the individual cell shape and what class or arrangement they were in. And don't worry, we're going to have plenty of practice so that you can become more familiar with this. So let's go ahead and let's take a look at some of the different epithelium tissues. All right, so our first one is going to be called simple squamous epithelium. So simple means that there's only one layer, and squamous is going to mean that it has like an abstract shape. Remember I said to you, a lot of people think it looks like fish scales under the microscope. So let's take a look at our illustration. Actually, let's start with B. And notice how once again, we're looking at a little slide. And as I mentioned to you before, when you look at your slides, oftentimes you're going to see multiple different tissue types. So you want to make sure that you take your moment and you figure out exactly where your tissue that you're looking at is going to be located. Now, when you look at epithelium tissue, what you can do is always look for an open surface. So this area right here, this is all open surface. And then that will help you identify your apical surface or layer of cells. So that means that these cells that are right here, this is going to be my apical surface. It's just one layer of cells. And I know that these are cells because as you can see right here, the nucleus has been stained for me. And I can see my little basement membrane. So I know that's where my epithelium ends. And looking at this, it just looks like one layer of cells, one string of cells. Now, what is their overall shape? So let's erase this real quick. Let's take a look. Now, I don't know about you, but I would say looking at their shape, each of them are slightly different. None of them look like little boxes or circles, and they definitely don't look like columns. So that means that this would be my best example of squamous. So since it forms one layer of cells, and since each cell has a uniquely different shape, we're going to call it simple squamous epithelium. Now, I hope as you're looking at it, you're going, well, what's happening over here? Why aren't we looking at these? Well, this part right here is actually muscle. So that part we're not interested in right now. Now, the reason we know it's muscle is because, well, I know it's muscle because as I can see, it doesn't have as many cells and it has more of a background. We haven't really discussed that yet, so I wouldn't expect you to know that just looking at this illustration. But that's also partly why on your left-hand side you have an actual slide, and then on the right-hand side on B, they've illustrated for you and highlighted which part of that particular slide sample is epithelium and which one is the rest of the different tissue types. Remember, epithelium tissue will always be the one that will do the covering. So if I come over to the left, it's going to be the one that's going to be exposed to the external environment. And as you can see, these are going to be my cells. They're forming one linear line. So it's a simple arrangement. And each cell has a uniquely different shape. It is not a square, not a column, not a circle. So that is why we're going to call it a simple squamous epithelium example. Take a look at the text that's below. It says it will involve a single row of thin cells. It is excellent for diffusion or transport of substances. And for our locations, as you can imagine, there are multiple locations within the body where you can find these particular tissue types. So to help you out a little bit, because I know for many of us, this will be the first time that we're really taking a look at histology, I don't want you to get too overwhelmed. So you're going to notice that on your PowerPoints, in the location, I'm going to go ahead and highlight some of them in blue. The ones that are in blue are the ones that I want you to study for your exam. These are the limit or the choices that I'm going to limit myself to ask you. So for instance, on your exam, I can say to you, what tissue type specifically can you find in the alveoli or the glomeruli? The glomeruli is in the kidney. And you're going to tell me simple squamous epithelium. Now, notice how you also find it in the endothelium and in the serosa, which is a layer, but notice how these are not highlighted in blue, which means is that I want you to read it, but I don't want you to worry about memorizing It's for its location. We're going to keep everything nice and simple, and we're going to look at the locations, only the ones that are highlighted in blue. All right, let's take a look at another example. This one is simple, so one layer. Cuboidal, so now we're looking more on a circular or box-like structure. Epithelium. So let's go ahead and take a look at what it says. It says it is a single layer of square or round cell. Once again, absorption and secretion, because that's what epithelium tissue does. For the location, I want you to know that you can find simple cuboidal epithelium in the liver, in the thyroid, and in the kidney tubules. In fact, the kidney tubules are the ones that are pictured for us on our picture above right here. Please keep in mind, as mentioned before, for the location, I'm going to limit my question types for the exam based on the ones that are highlighted in blue. So no liver, thyroid, and kidney tubules for simple cuboidal epithelium. And obviously make sure that you read the mammary and the salivary gland and the bronchioles. You can also find them there, but you won't have an exam questions on the ones that are not highlighted in blue when it comes to the location. All right, let's take a look at the picture. So as you can see, lots of different cells. How do I know where I'm looking for? Well, remember, find an open surface. Here's the little lumen. That's the little opening. So that's going to be all the cells that are exposed to the opening are in the apical surface. And I can see one layer of cells. Now, these cases, the layer is also able to make a little circular path. That's okay, because even though they're connecting, it's still one layer of cells. So it's a simple arrangement. And then next, take a look at the individual cell shape. And you're going to see that the majority of them are round or square. So that is then why it makes it a simple cuboidal epithelium. All right, how about this one? This one's pretty easy. So this one is simple, meaning one layer. Columnar. I think the columnar cells are the easiest one to find just because they're so elongated in their structure. So let's see over here. We're looking at a single row of tall, narrow cells. So here is my basement membrane. So that's where I know it ends. I have my open surface on top over here, and I can see that the cells are all lined up beautifully in one layer, and notice how they're very elongated. So you can see the nucleus has been stained. They're very elongated, so they're columnar in shape, one layer arrangement, so it's a simple columnar epithelium. All right, let's see what it says. Simple columnar epithelium, you can find them in the brush border of microvilli. They are often ciliated, and many of them possess goblet cells. Now, goblet cells I highlighted in blue because these are a particular cells that you're going to see over and over again in different chapters as we start learning more and more about the anatomy and physiology of the body. Anytime you hear goblet cells, I want you to think of mucus, because that is what they do. Goblet cells are secreting mucus for the body to use for a wide range of items. Now, location that we need to know for the simple columnar epithelium, we are just going to concentrate on the lining of the GI tract, the gastrointestinal tract, emphasizing that in the colon you find lots of simple columnar epithelium. All right, so hopefully as you're looking at your pictures for the epithelium, you're reminding yourself that the name comes from the individual cell shape and then the arrangement. So we've done lots of simple, meaning one layer. So let's go ahead and let's see if we can find some pseudostratified, or we can take some stratified epithelium and learn more of these different tissue types. All right, how about this one? This one is pseudostratified epithelium. Now, as I mentioned to you before, pseudo means appearing as if, so it's going to appear as if it's multiple layers, but it's only one layer of cells. So I'm going to go over here to my B, and I can see my basement membrane. I can see my apical surface, my open surface on top, so I know I'm looking at epithelium. And then what I'm going to do is I'm going to notice how the individual cells, even though they look as if they are stacked together, all of them are actually reaching the basement membrane individually. Because they're each reaching the basement individually, that really means that it's a simple arrangement, but it looks stratified because of the fact that it's almost as if you have more cells in there packed together than there should be in there. So it appears as if it's stratified, but it's really not. So this is pseudostratified epithelium. Now notice in the name how it doesn't tell you the particular shape of the cell, so it doesn't say pseudostratified squamous, pseudostratified columnar. The reason it doesn't have the cell shape is because oftentimes with pseudostratified, because there are so many cells squeezed into a small space, it's often very difficult to identify the individual cell shape. So that's why you can get away with just saying pseudostratified epithelium. Another way that you can easily identify this type is the fact that it's often ciliated. See how it has all the cilia that sits on top? So some of you, especially those of us who are doing the lab, might come across a tissue called ciliated pseudostratified. And that just indicates that ciliated means it had the fine little fuzzy hairs that helps you with mucus movement. It has that on top of it. All right, let's take a look at the text. So it says, it looks multi-layered, but all cells touch the basement membrane. You have cilia and you have goblet cells here in yellow. Mucus is secreted. Well, that's because your goblet cells are there. And for the location that we're going to need to know, the respiratory tract. So that's one of the orifices that we've picked because we know they're loaded full of pseudostratified epithelium. All right, now how about this one? This one's called keratinized, stratified. Stratified means 2 or more layers. This is supposed to be a 2. Squamous epithelium. All right, so let's start here on B. Now, because it says stratified, that means it's going to be multiple layers. The cells are all going to be very flat, abstract in shape, so we're going to call them squamous. So what does the keratinized indicate? We know what the stratified indicates. We know what the squamous indicates. What does the keratinized? Well, keratinized means that it's stuffed full of keratin, which often indicates that the cells are going to have a layer of dead cells that are stacked on top of the surface of your keratinized, stratified, squamous epithelium. And the keratinized indicator is where you know that you're looking for a layer of dead cells. Now, take a close look at picture B, and you're going to notice that you have your dead squamous cells that have been indicated for you. They're not going to stain as brightly as the rest because their nucleus is no longer functioning. And then if we look onto the right-hand side, you notice you're going to have your living epithelium cells. Notice how the living epithelium cells are all stacked together. The purple is the nuclei. So they're making full-on cell-to-cell contact. And because it has a layer of dead cells on top of it, that is then why it gets the keratinized label. And the layer of dead cells is actually very beneficial because it helps us with abrasion, and also it holds on to some water, so it reduces water loss from particular areas of the body where it has keratinized, stratified squamous epithelium. Probably the layer that most of us are familiar with are the bottom of our feet. Our bottom of our feet have a very thick keratinized layer of keratinized, stratified squamous epithelium. And in fact, if you ever go and get yourself a nice little pedicure, or even if you do a pedicure at home and you have to brush and you're brushing the bottom of your feet and you see all those little flakes falling off, that is nothing but the keratinized or the dead squamous cells flaking off, making way for the living epithelium cells to once again replace them. In chapter 6, we're going to discuss how the cells in epithelium go from living to dead. So we'll kind of study that particular aspect in our next chapter. For now, let's take a look, and we want to know that keratinized, stratified squamous epithelium can be found in the epidermis, which is the deeper layer of the skin, and by the palms and the soles. So these are excellent areas where you want to resist abrasion. So like for instance, the soles, because obviously that's what's touching the floor and any type of bottom surface. And then the palms are also excellent ways for them to fight off any abrasion. In case you fall, we often land on our palms or whatnot. So it's an excellent reason why we want to have that extra layer of dead cells to observe some of that shock. So that is where keratinized stratified squamous epithelium comes in. I don't think it should surprise anyone that if we have a keratinized, then we most likely also have one that has a non-characterized stratified squamous epithelium. So non-characterized means that you do not have a layer of dead cells. So all the cells here are living. They're all going to be stacked up in multiple layers, hence the stratified. And since we're looking at squamous epithelium, that means that they're going to have a relatively abstract shape when we look at them under the microscope. So let's go back over here to our illustration under B, and you can see nothing but layers and layers and layers and layers of cells just stacked on top of each other. Those are your stratified aspects. Notice how all of them are alive. It's making a connection with its basement membrane to the connective tissue. So that's the basement membrane and the basal surface. And then on the other hand, it is exposed to an open environment. So that is going to be the apical surface. Keep in mind that since all the layers, all the cells are alive, we're going to call them non-characterized stratified squamous, so no layer of dead cells. And for the location, it's going to be areas that are relatively moist. So the tongue, the esophagus, which leads us over to our stomach, and the vagina. Those are going to be our three locations where we're going to know that we can find lots of non-keratinized stratified squamous epithelium. All right, how about one that's cuboidal? So this one is going to be stratified multiple layers, 2 or more. Cuboidal, so once again, remember cuboidal means that it's going to have more of a box shape. It's going to look more circular. So we're looking at 2 or more cell layers, square or round for the individual cells. And if I look at B, I can see that I have one layer of cell here, and then I have a second layer that's coming around to it. So that's why it is stratified, and it's cuboidal because the individual cells do have a very circular or square shape. I think you can actually see it better in A than you can see it in B. But either way, for the location for the stratified cuboidal epithelium, we're just going to have one, and that's going to be the sweat gland ducts. Okay, so stratified cuboidal epithelium, you're going to locate that in the sweat gland ducts. And the name indicates that it's 2 or more layers and that the individual cells are squared or circular in shape. Now, our last epithelium is also one that doesn't really follow the rules of simple or stratified. This one is going to be called transitional epithelium. Now, transitional epithelium is going to indicate that we're looking at a multi-layer of cells. So here in my illustration, you can see lots and lots of layers of cells that are stuck together. Now, why don't we call them stratified? Well, it will all have to do with the apical surface or your top layer of cells. It turns out that the top layer of cells in transitional epithelium have the ability to change their size from round to more of an elongated, stretched out view. We're going to find this in areas that will give it a little bit of stretch. So for instance, in the bladder is composed of different tissue types. I told you that before. Organs have multiple tissue types. And obviously it's full of muscles because we know that the bladder will house our urine until we have time to go to the bathroom. But we also know that there's some epithelium tissue in the bladder, and the epithelium tissue will be transitional epithelium because its top layer of cells are able to go from circular, like in the picture, to more of an elongated shape. And when they elongate, they basically increase the lumen or the size of the bladder. So it makes it easier for you to hold onto your urine until it's time to go to the bathroom. So transitional epithelium, notice also how you don't have to tell me the individual shape of the cell. Transitional epithelium is going to be the epithelium that's going to have multiple layers, and its top layer is going to be able to stretch itself, thereby increasing the lumen that it is exposed to. So for its location, I have highlighted for you the ureter and the bladder. Now, I'm going to go ahead and stop here. This will be the end of the first category of tissue, which is the epithelium. And then in part 2 of our lecture, we're going to take a look at connective tissue, which is the most abundant and is the tissue that lies underneath the epithelium tissue that we've just discussed. And of course, we're also going to take a look at nervous tissue and the muscular tissue. As always, if you have any questions or comments, please feel free to email me, feel free to attend our Blackboard meeting sessions, and feel free to use the discussion board so you can interact with your classmates as well. All right, let's talk soon in the second part.